share my screen for y'all. All right. Can everyone see my PowerPoint? Awesome, perfect. So, like Susan said, my name is Mallory Gillispie, and I'm a second-year clinical perfusion student from the University of Nebraska Medical Center. And today I'm going to be talking to you about the effects of hemodilution on sonoclot parameters. And this was my thesis project along with my co-researcher, Sarah Pope. And we also had a lot of help from our director, David Holt. So first, just a quick outline of what I'll discuss today. I'll do a quick overview of the cell-based model of coagulation, as well as different coagulation monitoring tools that we use on bypass. Then we'll go over the Sienco Sonoclot device itself, and then we'll get into the nitty-gritty of our thesis research, as well as some conclusions we were able to draw and future avenues of research we could go down as well. So first, Brandon did a great job talking about the traditional coagulation cascade. However, there is a new way of thinking of coagulation as well. This is the cell-based model of coagulation, and it's similar to the coagulation cascade in that it has three general phases. So first, the initiation phase, which is, it begins at the activator phase on the diagram here, from activating factor 12 to 12A, 11 to 11A, and then 10 to 10A. And then we get that initial thrombin formation. So from that activator to that initial thrombin formation is our initiation phase of the cell-based model. Then the second phase is our amplification phase. And this is when thrombin has its own activators that it activates, so factors 8 and 5. And then the final phase of the cell-based model is the propagation phase. And this is what causes mass thrombin formation. So this is when those accelerators of thrombin, so activated 8 and 5, then further feed back to factor 10A, and we get that mass thrombin generation, which ultimately will convert fibrinogen to fibrin. And so while we're on bypass, it's important that we are monitoring our patient's coagulation status. And so the main indicator that we typically use would be our activated clotting time. And there are many, many factors that can influence a patient's ACT, whether that's hemodilution, temperature, the type of surface that you're using, coagulation factor deficiencies, as well as honestly, even the device that you use. And so some of the most common devices used are the Hemocron Junior Signature Elites, the HMS, the TAG, as well as the iStat. But Sarah and I decided to see how a different device compared to these. We use the Sienco Sonoclot, which is an ultrasensitive viscoelastic instrument. And so you select a reagent, and Sienco has created a very wide range of reagents for the entire hemostatic process. Some of the more common would be the Kalin or the C-Lite reagent. So you pick your desired reagent, you add a sample of whole blood to the cup, you place it in the chamber, and the pin oscillates up and down and generates the signature that you can see here as the three examples on the right-hand side. Another advantage to using the Sonoclot is the Signature Viewer software. It provides data collection, storage, as well as presentation tools and software that you can use after using their device. And this model connects really nicely with the cell-based model of hemostasis. So if we look at the top right diagram, we can see that it is highlighted as the coagulation reaction phase. This is what's going to correspond to our ACT. So this gives us that first initial thrombin formation. So again, this coagulation reaction phase is going to correlate really nicely with that initiation phase of the cell-based model. Then moving down to that middle diagram, there's an initial upslope that you can see, and that would correspond really nicely to the amplification phase of the cell-based model. So your thrombin accelerators are beginning to be produced. Then if you look at that middle picture still, the second phase of the sonoclot is the clot formation phase. This is when fibrinogen is converted to fibrin in forms of fibrin gel. So this would correspond to that final phase of the cell-based model, the propagation phase. And this clot rate is measured by a change in clot signal over a change in time. So this will vary depending on the patient as well as their overall physiological status. Then if you look in that final diagram, this is the third and final phase of the sonoclot signature, the clot retraction phase. And this is determined by the patient's overall platelet function, since functioning platelets are what help retract the clot. So the platelet function is given a score of 0 to 5. 0 meaning there was no clot retraction, and 5 there was strong clot retraction. So that's how we're able to measure a patient's platelet function using the Sienco Sonoclot. So for our research, we wanted to see with the use of the Sonoclot analyzer, a more accurate result will be obtained with use of details about the impact of hemodilution on various coagulation parameters, which can help assess the patient's status pre, post, as well as intraoperatively, and we can hopefully obtain a more accurate activated clotting time. And these pictures on the bottom are examples of the Sonoclot device itself, if you've never used it before. So our hypothesis was that if the patient's blood is hemodiluted on cardiopulmonary bypass, then it will increase the patient's ACT and clot formation time. So here's a review of the study method we used. Sienco was super generous with us and allowed us to borrow 12 Sonoclot chambers. And we were also really fortunate to be able to use our school's simulation suite to do all of our data collection, as well as even our subjects' whole blood collection process. We also measured the variables of ACT, clot rate, platelet function, clot signal, as well as time. And all those parameters are displayed really, really nicely on the actual Sonoclot chamber itself, as well as it connects nicely over to their signature viewer software. So here is a flow chart that we used for our experimental design. We drew 6 mLs of whole blood from each subject into 1 ml citrated vials, just so we didn't have any clotting going on while we're waiting in between trial runs. And then we divided these 6 1 ml tubes into all 6 of our hemodilution ratios. And so what we did was we had to reverse this citrate with 0.25 molar concentration of calcium chloride, which we got directly from the Sienco company as part of their protocols to reverse that citrate. And then from there, we had to pipette in the desired amount of plasmolyte to get our blood to plasmolyte ratio. So in normal volemia, we just used 330 microliters of whole blood. We ran those 2 tests on that. And then we had to pipette either a range from 220 microliters of blood down to 55 microliters of blood into each test tube with the appropriate amount of plasmolyte. So there was a lot of different size of pipettes and trial and error to get these correct dilutions in order to be able to run our tests. So the statistical methods we used a linear fixed model to model the outcome measurements of the varying hemodilution ratios. Our replicates were averaged within each of the 19 subjects for each condition, and we just did this in case there was an outlier of some sort, then we wouldn't have just ran 1 test per subject per dilution ratio. And we chose to compare each hemodilution ratio to all the other ratios rather than just to normal volemia. So we did take a penalty in our p-value for significance for doing this, but we thought it was important to compare the different ratios to each other and not just to a normal volemic condition. So here are our results. If you look at the graph on the right-hand side, the diamonds are going to represent the mean, and the line within each box is the median. And those clear little circles up top, those would be outliers if there were any for a given condition. So interestingly, we saw an actual decrease in our average ACT time between normal volemic and 1 to 0.5 blood to plasmolyte ratio. And then we did see that increase in activated clotting time as we became more hemodiluted, which is what we were hoping for and kind of expected to see. But what does all of this really mean clinically? What is this 1 to 5, 1 to 4 blood to plasmolyte ratio? So if we look at an average cardiopulmonary bypass patient, we'll say male, 75 kilos. We'll just say their prime volume was a liter with a pre-bypass hematocrit of 30 and just a 7% blood volume for body weight. If we calculate the dilutional hematocrit of solely the prime volume, so assuming no anesthesia fluids, no cardioplegia, no urine output, just simplifying this just for demonstration purposes, our prime alone would hemodilute that patient's hematocrit down to 25.2%. And this would correlate to a 1 to 0.19 blood to plasmolyte ratio. So if we take this patient and we look at the ratios that we used during our thesis research, you can see that we would have diluted our patient anywhere from a hematocrit of 20 down to 5. Now, we worked closely with John and Jane Henderson of Sienco to decide the ratios that we wanted to test. And we wanted to test the extremes of hemodilution to see how the Sonoclot could measure and analyze these dilutions. I hope we never give a patient 26 liters of plasmolyte on bypass, and I hope they have great kidney function and are able to, help us out there. But we wanted to just test those extremes to see really the impact of hemodilution on our ACT. So hopefully this table can give you a little bit of a better idea of what these ratios would mean in a clinical setting. So then looking at clot rate, you can see that we did initially see an increase in our clot rate as we diluted from normal volemic to 1 to 0.5 blood to plasma ratio. And then we saw a pretty consistent decline in our average clot rate. And this is what we kind of expected because your clot rate is the change in clot signal over the change in time. So as we diluted these patients, it took longer and longer for us to see those changes in clot signal. And so again, delta clot signal over delta time, if your denominator gets bigger, it's going to get smaller. So that's why we did see that decrease in overall clot rate. One thing we did find interesting was if you looked at just 1 to 2 and 1 to 4 blood to plasma ratio, there were no statistically significant relationships. So then the final parameter, it was our platelet function, and we saw a pretty consistent decline in overall platelet function as we increased hemodilution. This was originally not one of our testing parameters that we were focusing on. We were focusing mostly on clot rate and ECT. However, we were so impressed and just kind of surprised at the consistency of the impact that hemodilution has on platelet function. And as you can see in this chart, there was a very, very consistent decrease in overall mean platelet function as we increased hemodilution. So what does this all mean clinically? It means that we saw, with an increase in hemodilution, we saw an increase in average ACTs. We saw an overall decrease in clot rate, and we saw a decrease in platelet function. So these were just interesting conclusions that we were able to draw from our study. And some ideas of future research that we would like to take with our project would be to add heparin to the hemodiluted whole blood sample. So obviously on bypass, you need some sort of anticoagulant. So hopefully the next phase, for maybe our first years to come at Nebraska Med can take this and run with it, but would be to add heparin to our sample rather than just reversing that citrate from the initial test tube collection and running a whole blood sample. Another thing we would like to look at would be temperature effects. We kept our samples at room temperature from the time of collection to the time of the trial. So it'd be interesting to see, how does hypothermia and even hyperthermic states impact the sonoclot parameters. And then another avenue we would like to go down is platelet dysfunction effects. All of our patients were above the age of 18, healthy, no cardiovascular comorbidities, none of them were on platelet impacting medications. So presumably, all in all, healthy platelets. So since we saw this consistent effect of dilution on platelet function, it'd be interesting to see how just even minor platelet dysfunction or severe platelet dysfunction would impact this parameter. And as we know, on bypass, we are going to induce platelet dysfunction simply from just contact activation alone. And then a final thing we'd like to look at is the relationship between hemodilution and just platelet dysfunction in general, with or without the sonoclot. So here are some references. And I just want to say thank you to our statistician, Kaylee Sampson, our professor, Scott Sanderson, our classmate, Mike Plymell, who was a phlebotomist before he was a perfusion student, and he donated all of his time to collect all the blood samples for us, so we really appreciate it. Also, John and Jane Henderson at Sienco. They were so helpful and so generous throughout this entire process. And also, thank you to the Sanibel Symposium audience and team who put on this meeting. I really appreciate the opportunity to talk to you guys today. Thank you.
Thank you.